Hello, welcome to my analysis of how DNA is broken down and copied to create other chromosomes, creating other cells from there, programming and creating the human body and its unique DNA structure. DNA is amazing. Each cell in our body contains about 2 meters or 6 foot of DNA if it is stretched out. But how can we fit this much into one cell? Well, this starts when protein molecules called histones attach to the edge of the DNA double helix and begins to coil the DNA around them. This is called nucleosome, and each of these is then packed into one compact strand. This is known as a chromation. Now this strand is again coiled and twisted into a spring-looking shape. These spring-looking shapes are called solenoids, and they are finally twisted to become chromosomes, which we can see through a microscope. Breaking down the DNA strand we started with, we find that this is made of 2 coiled strands known as a double helix. Unzipping these strands, and we find that each one has a long sequence of the letters A, C, G, and T spread along it. This is its genetic code. Each side of the double helix is an identical match, as in the code an A must always be opposite to a T, and a C must always be opposite to a G. This means that we know both sides of the DNA while only reading one. The order that these letters are aligned in is the unique genetic structure and code of each person's DNA, and what makes each person different. DNA must be copied for cells to duplicate and for the body to grow. This process happens when a group of proteins gather around the DNA double helix and form some sort of machine which places another protein over the DNA and shoots it down the DNA strand. As this protein moves down the DNA, it unzips it and makes a yellow and green strand to start to appear and grow out of this protein. This strand is called RNA and is a direct copy of one side of the DNA. This RNA is made of introns and exons. The exons are the part of the RNA containing the code, and the introns are not needed in this process. So there is a process known as splicing that is used to take out these intron sections. You can see this happening right here. What is happening is a protein is forming around the introns and then cutting it out and then rejoining the exons to make a finished RNA strand. The RNA then goes to the outer areas of the cell. Now, similar to the protein that unzipped and copied the DNA, another protein assembles around the RNA. This is called the ribosome. It moves along slowly, taking in small proteins, then encoding and separating them out into a final strand. This is a protein that has an exact copy of the genetic structure of the person. As we can see, all DNA in the body is exactly the same and is very complicated, so if somehow this DNA pattern in a certain part of the body is to change, it can cause problems, and this leads to mutations and diseases like cancer. For example, when someone gets burnt, their skin has been exposed to too much UV light. This makes small sections of damage to parts of the DNA. This is usually patched over by the body, but in some cases, if it's bad enough, it can cause deformity to grow, and this spreads to cause mutations and diseases.